This UCSD TV program is presented by University of California Television. Like what you learn? Visit our website or follow us on Facebook and Twitter to keep up with the latest programs.
We are the paradoxical ape. Bipedal, naked, large-brained, long the master of fire, tools, and language, but still trying to understand ourselves. Aware that death is inevitable, yet filled with optimism. We grow up slowly. We hand down knowledge. We empathize and deceive. We shape the future from our shared understanding of the past. Carta brings together experts from diverse disciplines to exchange insights on who we are and how we got here. An exploration made possible by the generosity of humans like you.
So I will discuss today the relation between inflammation and metabolism and metabolic homeostasis and disease susceptibility. So inflammation is a protective response to a variety of challenges that we may experience, including infection and injury and various types of tissue stress and malfunctions and all the things that can go wrong. All of them can induce an inflammatory response that meant to be protective. And depending on the type of challenge that we experience, there's different physiological or intended purpose of the responses, such as host defense from infection, tissue repair response, or restoration of a normal state of the tissues. And what is very interesting about inflammation and very clinically important is that all these different pathways to inflammation can result in a variety of pathological sequelae, including autoimmune diseases, sepsis, fibrotic diseases, tumor growth, and a variety of diseases of homeostasis that are becoming more and more common in modern lifestyles. To give a very, very simplified summary of inflammation, of inflammatory pathway, when inducers of inflammation, such as pathogens or tissue damage, are encountered, cells in our body, such as macrophages and epithelial cells and so on, detect these inducers and produce various inflammatory mediators, including cytokines that some of them were mentioned already. And what these mediators do, they act on practically every tissue in the body, and they change some functional characteristics that intend to either cause elimination of whatever caused inflammation or adaptation to the presence of these conditions. And why inflammation is so broadly associated with diseases, very symbolically, it could be summarized here. So for every trait, including defense traits, there are benefits and costs. For defenses, the costs are particularly pronounced. And the traits would be evolutionarily stable if the benefits are higher than the costs. And anything in this green part of the triangle would be therefore potentially evolutionarily stable, and everything here would be unstable. And the problem is that in the case of traits that can provide very high benefits, such as survival, the acceptable cost can also be very high. And that leads to this type of picture where the part of this upper triangle that's on the right side, where benefit can still outweigh the cost, but in terms of the absolute value of the cost, this would be already, from the patient perspective at least, and the doctor's perspective, this would be already in the disease zone. These would be conditions that we would experience as something that is definitely not well-being, and doctors would diagnose as being various types of pathological or disease conditions, even though they are still provide more benefit than the cost. And what we are interested in is in the biology of this upper right corner, where the high benefits associated with very high costs, and where we basically on the edge of the chaos or transition into the pathological states that can be potentially lethal. And this type of biology deals with not just common conditions like mild infections or mild anomalies, but it has to do with essentially biology of survival. What are the kinds of mechanisms that are employed by our system, by our organism, in order to survive critical conditions, the ones that are just one step away from death. And this is just in a very schematic way summarized here. So if we consider this type of a disease tree, there are many conditions that we can are one step away from health. These are various common mild illnesses. These are conditions that make you go to see a primary care physician. If there are complications of these conditions, you get referred to some internal medicine specialists. These are more complex diseases. But you can still recover and go back to health. And in severe cases, you can get into this critical illness state when you get into ICU. And you still can recover from here. And in this critical illness state, which is conditions like sepsis, pneumonia, heart attack, brain damage, and so forth, in these conditions, you are one step away from these three proximal mechanisms that lead directly to death. And these are points of no return, which is respiratory failure, cardiovascular failure, or damage to the brain areas that control respiration and cardiovascular function. So we're interested in what happens here, what kind of mechanisms may be evolved to prevent this transition into this point of no return. The case I will discuss today has to do with this very enigmatic and very interesting and very familiar to everyone set of conditions known as sickness behaviors. This is something everybody in this room experiences when you have flu or any other type of severe infection or other types of acute illness. We all experience this set of conditions such as loss of appetite, social withdrawal, fatigue, sleepiness, and so on and so forth. So these are all stereotypic conditions that are associated with acute illness, most commonly with acute infections. So flu symptoms would all be in that category. And the biology of sickness behavior has not been really well understood, although there were early studies that demonstrated that sickness behavior is not just a consequence of system being destroyed by a pathogen, but rather these are motivated behaviors. In other words, they are intended by the organism for some reason. And there's been many speculations that whether various aspects of sickness behavior perhaps help the immune system to fight the infection, or maybe there is some other way that they may contribute to dealing with acute illness. And so we were interested in investigating aspects of acute illness, including loss of appetite and change in sleeping patterns, and how they may contribute to survival of the acute illness. So there are two ways that we can survive an infection or any other challenge for that matter. We can either resist it. So in the case of infection, normally we would be in this state where we are healthy and there is a low or a negligible pathogen load. And as we get infected and pathogen expand, we can become ill, as indicated by this position. And from here, we can either go back to health by getting rid of pathogen, which is the role of the immune system, and this is referred to as a resistance to disease, or we can adapt to the presence of the pathogen and go back to the healthy state despite microbial load, and this would be referred to increased tolerance to infection or tolerance to damage. There are two ways that pathogens cause damage. One is directly through tissue destruction, through toxins, virulence factors, and so forth. And the second, more common way the pathogens, infections called illness is due to damage by the inflammation caused by infection. And so we were interested in figuring out whether sickness behaviors provide benefit, first of all. Secondly, whether they provide benefit because they promote immune function and resistance to infection, or whether they provide benefit by promoting tissue protection and tolerance to inflammatory damage or pathogen-induced damage. So just like in humans, in mice, or in any animals that's been studied, all the way back to insects, when they are ill with acute infection, they stop eating. This is food consumption in mice after they received a dose of listeria infection. This is a common food poisoning type of infection. They stop eating and until they start clearing infection and they start recovering, and then they start eating again. But there is a very profound anorexia associated with infection, as you can see here. So some studies done over 40 years ago actually found that in the same model of listeria infection, if mice are force-fed, then mortality increases. And this is what we reproduce here. So this is showing survival of mice that are given LD50 dose of listeria, dose that kills half of mice. And this is what's shown here. And if they're force-fed, then all of them die. And they're fed just 20% of daily caloric intake. So they're not really stuffed with food. They're just given a little bit of food, and that kills them. And the food we use is actually the same food that's used in ICU units. And then we asked what component of food kills. And so we gave them separately proteins, fat, and sugar. And turns out that sugar alone or glucose alone was sufficient to kill them, whether it was given orally or intraperitoneally. So just giving them a small dose of sugar, only 2% of normal daily caloric intake of glucose, was sufficient to kill 100% of mice. And conversely, if we gave them glucose inhibitor called 2-DG, 2-deoxyglucose, it's a modified form of glucose that cannot be metabolized, so it blocks glucose utilization, then 100% of mice would survive. And importantly, this was not because the immune system was affected in its ability to get rid of the pathogen, and it's not because there was higher dose of higher magnitude of inflammation, because these were not different between mice given glucose or not, as you can see here. So this indicated that mice survived or died from this manipulation because of the effect on tissue protection, not on the immune function itself. So then we asked what would happen in a more severe condition like sepsis. Sepsis is the condition when infection becomes systemic, when it gets into blood. In this case, it's a bacterial sepsis that's mimicked by giving mice this component, endotoxin or LPS. And as you can see here, if mice given LPS or septic mice, if they're given food, they die, compared to control where they're just giving saline solution. And importantly, if they're just given sugar, then they all drop dead. But most excitingly, if they're just giving the simple drug that blocks glucose utilization, then 100% of them can be rescued from sepsis. And sepsis is a really terrible, intractable condition that is still a very common cause of death in ICUs. So again, the mortality or protection from death in these manipulations was not dependent on the degree of inflammation, as shown here with example of a couple of inflammatory mediators. And it again indicating that it was due to increased tissue protection from damage caused by severe inflammation that is associated with sepsis. Then we asked what would happen in a different type of infection. So far it showed you bacterial infection and bacterial sepsis. And now we asked what would happen in influenza virus infection. So when mice are infected with flu virus, again, they undergo this period of anorexia when they stop eating until they start recovering. And if they're force-fed, to our great surprise, they actually survive better. So this is now giving almost lethal dose of flu infection. Most mice die, but when they're giving that food, most of them survived. And also, if they're giving glucose, that also provided partial protection. And if they're given 2DG, glucose inhibitor, then all of them dropped dead. So it was exact opposite to what happened in bacterial infection and bacterial sepsis. And again, the effect of glucose was not due to its impact on inflammatory response. So in viral infections, it's a different type of inflammatory response, which is dominated by interferons, shown here. They're the same. And the dose burden of the virus in the lungs was also the same, was not different. Again, suggesting the effects are due to impact on tolerance to tissue damage. And the data that I'm not going to show, but I summarize here, what we found is that death from viral inflammation was associated with decline in vital functions, like respiration, blood pressure, and so on, suggesting that there was a failure in autonomic control centers in the brainstem. And if mice are given food or glucose, then these declines in autonomic functions could be rescued. If they're given 2DG, they're enhanced. In contrast, death from bacterial sepsis was associated with neuronal damage in the midbrain area, and it was also, the death was immediately preceded by convulsions. And these were blocked by 2DG, and they were enhanced by glucose. So we did PET scan analysis and found that glucose utilization under conditions of bacterial inflammation versus viral inflammation was very different, and it segregated exactly to these two brain areas, such that in viral inflammation, it was primarily in brainstem, and in bacterial inflammation, it was in the midbrain area. So, and the delivery, increased delivery of glucose to these areas related to the impact of glucose or glucose inhibitors on the damage to these areas. And then we asked what is the mechanism of damage and what's happening there. So I will just summarize that part of the study. What we found was that in the case of viral inflammation, what happens with cells that are infected with the virus, one of the things that happens, there is a particular type of cellular stress response known as unfolded protein response that has two branches. One branch tries to adapt to this stress condition and to resolve it. And if that fails, the second branch would kill the cells as a second option. And this death from this response is mediated by this particular gene called CHOP. It's a transcription factor. So then we asked whether, and what is known is that glucose deprivation can exacerbate unfolded protein response, this stress response pathway. And so we hypothesized that that brainstem area, the neurons in the brainstem, for reasons that remain mysterious, underwent this excessive unfolded protein response stress that was ameliorated by glucose and exacerbated by 2DG. And then we asked whether this pathway of cell death or neuronal damage mediated by this gene is involved. And for that, we asked whether mice that have a mutation in this gene, whether they would be rescued from the effect of such inflammatory manipulation. And indeed, what we found, if we just look at this blue line, these are mice that have infection, that have viral inflammation, and they're given 2DG, they all die. And if they don't have that gene, CHOP, then they all survive. And so the mechanism here is related to blockade of this pathway for neuronal dysfunction. And finally, in the case of sepsis or bacterial inflammation, fasting metabolism that protected them from death was related to the switch from normal fat metabolism to fasted metabolism, which is there's a switch to fuels from glucose to ketones. And that switch was necessary for mice to survive sepsis. And this is shown here, that giving glucose blocked transition to fasting metabolism, and deletion of the gene that is responsible for ketone production made mice susceptible to sepsis. And what, as I mentioned, immediately before death, mice had convulsion in sepsis. So what we did in this final set of experiments, if we gave the mice anti-epileptic drug called valproic acid, then we could rescue them from death even when they're given glucose. And so that indicated that the reason that that worked is because valproic acid, unlike this other drug shown here, which is Keppra, which is also anti-convulsion drug, valproic acid, one of its mechanisms is related to the effect, to the same effect that ketones have on a particular class of enzymes in the cell. So I will summarize it here, that there are two different pathways to death, and fasting metabolism is protective in one case and detrimental in the other case. And the implication for human treatment in ICU is that all clinical trials done so far with nutrient manipulation in ICU units have been done on patients that have not been separated based on cause of sepsis, bacterial versus viral, and the results of these studies were mixed. And we think that it's because they were not separated and they basically canceled each other out. So now we are planning to conduct a clinical trial where we'll separate them based on cause of sepsis. And so this is the summary. And finally, this work was done by three very talented scientists in the lab, Harding Luan, Sarah Huyen, and Andrew Wong. And we had help from other colleagues at Yale. And thank you for your attention.